we are the Vietnamese team, and our group consists of 4 members, including Lê Thiều Hân, Phạm Quốc Anh, Hoàng Xuân Tùng, and Vi Lê An Bích Phương. Today we'll present to you our research about the effect of mimosine from Vietnamese Mimosa pudica in carrageenan-induced arthritic mouse model. Rheumatoid arthritis is an autoimmune disease in which the body's immune system attacks joint tissues, causing inflammation in the joint lining, leading to cartilage degradation. So, our treatment objectives are to reduce inflammation, inhibit the pattern's formation and morphological variation, and provide long-term pain relief. The drugs included in our experiment will reduce the biological mechanisms which are involved in the inception of inflammatory response. So the material that we are examining is mimosine, which is an active component of Mimosa pudica. The pharmacological activities of mimosine have been demonstrated in vitro and in vivo. So our aim is to isolate the mimosine from the Mimosa pudica and investigate the role of mimosine during the regulation of the inflammatory signaling in macrophage and the anti-inflammation of mice with carrageenan-induced arthritis.
Hi, my name is Han. In figure 1, microphage cell were isolated from mice. Then the cell were cultured in BMEM supplemented with 10% ABS. The displayed macrophages were incubated at 37 degrees Celsius with 5% carbon dioxide. Moving to figure 2, displayed the isolation of mimosine from Mimosa pudica. In the figure 3, to determine the concentration of capsaicin to the viability of macrophages. Macrophages were divided equally into different wells on 96 were placed at the destiny of 1 multiplied by 100,000. These cells were incubated with mimosine at 10, 20, 30 microgram per milliliter for 24 hours. Then the cells were incubated together at 20 microliter cell titer 96 aqueous 1 solution cell assay system technical protein. From the result, we found that at 30 microgram per milliliter, mimosine was not toxic to the cells.
Hi, I am Phạm Quốc Anh. In figure 4, to examine whether mimosine can affect the RPS-induced production of pro-inflammatory cytokines. Mimosine concentration 5, 10, 20, 30 microgram per milliliter were examined their effect on pro-inflammatory cytokine production of interferon gamma. The results of the figure 4 indicated that the release of the IFN gamma was strongly inhibited by macrophage at a concentration of 30 microgram per milliliter. In figure 5a, after 30 minutes, mice dry food was injected with 0.05 milliliter carrion to 2%. Then it was reddened, and there were local inflammatory symptoms. According to figure 5a, the uninfected mouse is not inflamed. In figure 5b, the mice are also inflamed after using mimosine at 30 mg per kilogram. In figure 5c, the thickness of the dry food of mice was significantly reduced. In particular, at a concentration of mimosine at 30 mg per kilogram gives the highest therapeutic effect. After 2 hours using mimosine on group, a mice was treated with mimosine reduced swelling, and after 24 hours, and the swelling of the joint was almost normal compared to the left leg without inflammation. Inflammation of the CMC injection group and the white group after injection carrageenan increased over time.
Hello, my name is Hoàng Xuân Tùng, and in figure 6 right here, we have 2 pieces of the ears of mice. The right one has dimethylbenzene in it, and it's clearly swollen. It has some kind of reddish color to it. But the left one is completely normal because it has no dimethylbenzene in it. So yes. Moving on to the figure 6b, we can see that the differences in weight between the right ear with dimethylbenzene and the left one, the non-inflamed one, varies among the groups. Specifically, those differences in the mimosine involved groups were all vastly lower than those of the control group. And the mimosine at 30 mg per kilogram has the best effects in alleviating the inflammation. Moving on to the table number 1. In order to assess the effects that mimosine has on the weight of these mice, we performed a weekly mice weight, and at the very same time we also observed their expressions of eating, drinking, and mechanical movement. And based on that data, we can say the mouse weight increased, there was no major difference in the weight between the groups, and the mice in the treatment groups did not show any kind of abnormal activities compared to the control group. So what might be our conclusions here? The first one is that the mimosine of 30 microgram per milliliter inhibited the IFN gamma levels in the macrophage cells. Next up, the in vivo mimosine of 30 mg per kilogram inhibited the rheumatoid arthritis. All in all, the results of our study demonstrated the mimosine improving the rheumatoid mice, which sheds light on a novel strategy in treating the rheumatoid arthritis.
Thank you for listening, and we hope you enjoy our presentation.
Thank you.